Thank you very much for having me today. I'd like to talk to you today about our use of Python in our imaging lab inside the Mayo Clinic. And for those of you who may not be aware, Mayo Clinic, just a brief spiel, largest private not-for-profit group medical practice in the world. We treat about 1.3 million individual patients each year in the last year from 135 countries around the world. 120 million Americans are in network, and the organization is 150 years old this year, which is quite nice. And we're also proud to have been recognized as the best hospital in the USA for 2014 and 2015 by US News and World Report. So thank you very much to the Python community for developing all these powerful tools that are behind everything that I'm going to show you, pretty much everything that I'm going to show you. Without it, it would have been a lot more difficult, and we wouldn't have made as much progress as we have, as quickly as we have. So I'm going to talk to you today about Alzheimer's dementia. That's the particular form of dementia that we're interested in. It's an incredibly costly disease. Last year is estimated to almost $230 billion were required to treat patients with Alzheimer's in the US, and nearly 2/3 of this was shouldered by Medicare and Medicaid. As you can imagine, the patients tend to be elderly. There are 5.3 million people in the US who have a diagnosis of Alzheimer's, and 2/3 of those are women. And what's really scary about this is where this is headed. The estimates claim that by 2050, those numbers will have tripled. Will be around 16 million people with a diagnosis, and the cost could bloom to over $1 trillion a year. And this will bankrupt Medicare and Medicaid if something isn't done and done soon. There's just no way that these two agencies can possibly fund this. Someone mind closing the door? The other thing a lot of people don't realize is that Alzheimer's kills. It's not just a disease of impaired memory. It is quite vicious. If we look at a plot here of a lot of the major sources of mortality in our disease, breast cancer, prostate cancer, and look at the percent change in death over the years 2000 to 2013, it's quite interesting. Most of the major causes of death have dropped dramatically. Breast cancer, prostate cancer, even stroke is down quite a bit. But over the same period, if you look at Alzheimer's, it's an alarming increase as the cause of death in Western society. In fact, it's now the 6th leading cause of death in the US. And what's perhaps most amazing about it is that it cannot be prevented, cured, or even slow. And so this drives the need, both the economic and the human toll of this disease. We need a non-invasive imaging-based biomarker so that we can measure the brain's response to new drugs in clinical trials. And we hoped, current thought is that we need to start treating this perhaps 20 years before the patient appears with symptoms. So the earlier we get on the brain, the better. So one of the interesting things about Alzheimer's is it has a hallmark pathology and progression. The beta amyloid plaques you may have heard about in the tau tangles, it starts in the entorhinal cortex, moves into the hippocampus, and then from there proceeds broadly throughout the brain. And what we're really interested in is the earliest stages of this disease. These structures are involved in memory formation and recall in general. And so this hallmark pathology and hallmark progression that differentiates this from other forms of dementia is something we want to leverage. This drives the hypothesis of this research, which is that ongoing Alzheimer's pathology will cause structural changes in these regions of the brain, and that we can quantify these using high-quality MRI and some sophisticated texture analysis algorithms. So to test this out, we decided to conduct a short pilot study before we go and invest large efforts into recruiting large numbers of patients. We looked to a publicly available, commonly used data source for Alzheimer's research called the ADNI dataset, Alzheimer's Disease Neuroimaging Initiative. And this is a longitudinal study that started in 2004, 11 years ago now. And it collects imaging, cognitive data, and tissue biomarker data from a large number of patients. The 3T MRI is performed. That's a high-quality MRI, relatively high field strength for medical imaging. And 3T MRI images are performed at the screening exam, baseline, 6 and 12 months. There are 800 patients enrolled in ADNI-1. I'm going to keep talking until they tell us to leave. 200 normal cognition subjects, 400 with mild cognitive impairment, and another 200 with full-blown clinically diagnosed Alzheimer's. So we don't have a direct measure of pathology in the brain, so we have to use a surrogate. And the surrogate we've decided to use is a scale called the Clinical Dementia Rating, or CDR for short. It's a 5-point scale characterizing things like the subject's memory, their spatial orientation, problem solving, personal care habits, and social interactions, things like that. This is collected in the clinic through a structured formal interview of both the patient and their caregiver and family. You can see the 5 points in the scale there, so increasing value of the rating corresponds to increased impairment of memory and cognitive function. And 0.5 is questionable. That's sort of a gray zone. We know they're not normal, but we wouldn't quite classify them as having mild cognitive impairment yet. So we looked at a pool of ADNI data that was available on the website and had a sample size of convenience. Basically, we looked for 10 people in each of these groups. It turned out, we looked at the 6-month exam, there were no severe patients in this group, there weren't any available, and only 9 in the moderate group. But it was what it was, so we went with 39 patients or subjects in this study. And this is an example of what the MRI looks like. This is a 3T scan of the brain, and this is a coronal acquisition. If you're not familiar, coronal is slices this way from the face working backwards. Axial is this way, and sagittal is this way through the brain. So this is a coronal slice, and it's preferred for studies of the hippocampus because it's readily visible in this orientation. So these studies have typically 256 images crossing the whole brain. This is a T1 weighted non-contrast slice. We selected 3 of these images that cover the hippocampus in both the left and right hemispheres, and the little yellow boxes indicate the regions of study that we're interested in. Those are the hippocampi. So we had a human expert, a neuroscientist, place 16 by 16 regions of interest over both the left and right hippocampi. And the goal was to calculate texture features in these ROIs and average them across the 3 slices and produce sets for both the left and right hemispheres. And part of this is because the pathology is known to progress differently in the 2 hemispheres. We didn't want to pool the 2 together. So, and this data, the imaging data, neuroimaging data, and the regions of interest were fed into our, what we call our Python texture analysis pipeline. And it's a growing set of highly cited or previously useful texture analysis algorithms. And I've listed some of them here, sorted by year. So the gray level co-occurrence matrix is the oldest of these. It's been cited over 11,000 times in the literature, so broadly useful in a wide range of applications. And we've implemented a bunch of others. The ones at the bottom are relatively new. The last one, the Laplacian of Gaussian, is the basis for a commercial product that's quite popular. The discrete orthonormal Stockwell transform is something that we developed in my lab. And it's a combination, it provides combines some of the nicest features of Fourier and wavelet transforms. It's a multi-scale Fourier analysis, very powerful for a lot of applications. So as you might imagine, processing all these pixels and all these slices lends itself quite well to parallelization. We've done this so far. We used a parallel IMAP to parallelize across cores and some initial experimentation. This is on an 8-core iMac. It performs quite well. I was quite surprised at how well Python scales with cores for this particular application. And when you get into the virtual cores, it looks like it's really recovering something there. And when we have all the hyper-threading turned on, it looks like we're getting pretty close to our theoretical performance limit. It's quite nice. We're going to extend this further in the future to parallelize across slices in a cluster mode, perhaps. We're looking into it now. So the output from this texture analysis pipeline is a set of texture features per patient or per clinical dementia rating. So you can think of this as a row in a spreadsheet. And if we dig a little bit deeper into these texture features, what we've done is we've grouped them together by the left and the right hemisphere, the left and the right hippocampus. And then within each of these structures, we have the features generated by each of the algorithms, plus some first-order features that apply to the region as a whole. So this is essentially what we're going to use to build and test our model. But if we look at this, we have 155 texture features per subject. That's what we get out of these algorithms. And that's way, way more than the number of subjects in the study. So we've got some issues. So what we, first thing we do is reduce the features through feature selection, and we use a couple of algorithms. It ranks by correlation with the CDR. We pick the top 20 features and pass those through into a best first algorithm, which looks for the features that are least correlated and comes up with a final set. Once we come up with a final set, that's passed on to the next stage of the experiment. We conducted 4 experiments, doing pairwise comparisons to see in this pilot study if we could detect any difference at all between any of these levels, because it hasn't been done before, and we weren't sure that there would be any kind of detectable difference between these in the structure of the hippocampi and these clinical scales. So we used multiple classification algorithms, as we often do, and did a leave-one-out cross-validation in each case, typically a 20-fold leave-one-out cross-validation. So these are our preliminary results. We got pretty good results ranging from between 74 and 94% accuracy in these 4 experiments. And it turned out that the feature selection algorithm chose typically 3 or 4 of the original features. These are not linear combinations of the original features that you might get, for example, out of a PCA type of analysis. These are just the original features. And in every case, less than 12 features of the original features were selected. And the accuracy numbers I'm showing are from a naive Bayes classifier, but it was pretty typical of the ones that we use, certainly in the ballpark. So in summary, this is the first report that we know of that correlates structural MRI, anatomical MRI, and a scale that's used frequently in the clinical setting to measure dementia. And we're able to differentiate this clinical scale, different clinical scales in these patients with a reasonable accuracy given the small size of the study. And importantly for us, the low values in this scale, we were able to differentiate 0 and 0 and 0.5 and 0.5 and 1 with the 85% accuracy. We're really interested in the early stages of the disease in particular. And also surprisingly, it required a small number of texture features. We're very aggressive about feature reduction so we don't overfit the data. And we're quite happy with that results. However, lots of caveats. It's a pilot study, so we have a small N. There's a large pool of data that we can collect from, so we can enlarge this in the future. This was done to see if there was any value in going to that extra work. And we perform multiple pairwise comparisons, which is generally a bad thing. We should be doing an ensemble comparison across all of them unless we have a specific reason to only want to look at a particular pair. But again, a pilot study to see if we could detect any differences at all. And we only had 1 anatomical MRI data set. Patients in the clinic, if they came in now, would get a much broader and richer array of imaging, functional and anatomical and physiological imaging performed, and we could incorporate this in our clinical studies. Nevertheless, the results are encouraging. We will go on and collect some more data from both ADNI and hopefully in the clinic as well. And the Python and the packages that are out there helped enormously. We probably wouldn't have been able to do it without all the fine effort that you folks have contributed. Thanks very much for your time. So it's a leave-one-out cross-validation. So you train on, let's say, 19 and test on the 20th. Do that 20 times, and we know the truth. That's how you get an estimate of accuracy, sensitivity, specificity. Yes?
Do you have any sense for what the discriminative features refer to? Is there any biological?
So the question is, do we have any sense of what the features mean biologically? No. All we can say at this point is that they tend to, there's a wide range of features. These algorithms generate a lot of different things. But we did do experiments where we looked at features over a broader and a smaller spatial scale. And the broader scale, about on the scale of the hippocampus, were the ones that gave us the most information. And there's actually, all the information in the smaller scale is also in the bigger scale. So that sort of makes sense. There's more information in the larger, but constrained to be within the hippocampus. So it's something about the structure of the hippocampus that's, and remember, this is not readily visible in the MRI. To a radiologist looking at these scans, they can't tell whether the patient has dementia. But the texture is finding something that's there. Yes? So the question was, are we going to apply this to people who do not have a diagnosis but have one of the genetic markers that makes them highly predisposed to get it? Absolutely, that's the goal ultimately. And we're working very closely with neuroscientists at Mayo Clinic, and we have a large practice in that. Yes.
How big were the differences in accuracy for different classifiers?
They all generally range. We found that the plain tree classifier performed by far the worst. Random forests were about the same as the naive Bayes, and things like SVM are right in the ballpark too. So not a lot of difference, 5–10% between the different classifiers. And we do that to sort of reassure ourselves that we're not overfitting. Yeah. Yeah.
There's time for one more.
Yeah.
So if I'm understanding right, the feature selection was sort of done individually for each pairwise comparison.
That. Go ahead, finish.
So the question is, are similar features coming out as they progress, or does it look like it's different features changing?
So the question was, first part, did we do feature selection for each of those 4 separate experiments? The answer to that is yes. That was done on a per experiment basis. That's part of the problem with multiple pairwise comparisons. And then the second question was, were similar features coming out? And there was some similarity in the features, but as there were unique features for each different experiment, but there were some that were in common. And surprisingly, the discrete orthonormal Stockwell transform was fairly commonly used, as was the local binary patterns. Those tend to be selected a lot.
Okay, well, let's thank the speaker again.
Thank you.